Section number 1 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. Structure of the Nervous System Part 1. The nervous system is the most complicated and highly organized of the various systems which make up the human body. It is the mechanism concerned with the correlation and integration of various bodily processes and the reactions and adjustments of the organism to its environment. In addition, the cerebral cortex is concerned with conscious life. It may be divided into two parts, central and peripheral. The central nervous system consists of the encephalon or brain contained within the cranium and the medulla spinalis or spinal cord lodged in the vertebral canal. The two portions are continuous with one another at the level of the upper border of the atlas vertebra. The peripheral nervous system consists of a series of nerves by which the central nervous system is connected with the various tissues of the body. For descriptive purposes, these nerves may be arranged in two groups, cerebrospinal and sympathetic. The arrangement, however, being an arbitrary one, since the two groups are intimately connected and closely intermingled. Both the cerebrospinal and sympathetic nerves have nuclei of origin, the somatic efferent and sympathetic efferent, as well as nuclei of termination, somatic afferent and sympathetic afferent, in the central nervous system. The cerebrospinal nerves are 43 in number on either side, 12 cranial attached to the brain and 31 spinal to the medulla spinalis. They are associated with the functions of the special and general senses and with the voluntary movements of the body. The sympathetic nerves transmit the impulses which regulate the movements of the viscera, determine the caliber of the blood vessels, and control the phenomena of secretion. In relation with them are two rows of central ganglia, situated one on either side of the middle line in front of the vertebral columns. These ganglia are intimately connected with the medulla spinalis and the spinal nerves, and are also joined to each other by vertical strands of nerve fibers so as to constitute a pair of knotted cords, the sympathetic trunks, which reach from the base of the skull to the coccyx. The sympathetic nerves issuing from the ganglia form three great prevertebral plexuses which supply the thoracic, abdominal, and pelvic viscera. In relation to the walls of these viscera, intricate nerve plexuses and numerous peripheral ganglia are found. Structure of the nervous system. The nervous tissues are composed of nerve cells and their various processes, together with a supporting tissue called neuroglia, which, however, is found only in the brain and the medulla spinalis. Certain long processes of the nerve cells are of special importance, and it is convenient to consider them apart from the cells. They are known as nerve fibers. To the naked eye, a difference is obvious between certain portions of the brain and medulla spinalis, that is, the gray substance and the white substance. The gray substance is largely composed of nerve cells, while the white substance contains only their long processes, the nerve fibers. It is in the former that nervous impressions are received, stored, and transformed into efferent impulses, and by the latter that they are conducted. Hence, the gray substance forms the essential constituent of all the ganglionic centers, both those in the isolated ganglia and those aggregated in the brain and medulla spinalis, while the white substance forms the bulk of the commissural portions of the nerve centers and the peripheral nerves. Neuroglia. Neuroglia, the particular ground substance in which are embedded the true nervous constituents of the brain and medulla spinalis, consist of cells and fibers. Some of the cells are stellate in shape, with ill-defined cell body, and their fine processes become neuroglial fibers, which extend radially and unbranched among the nerve cells and fibers which they aid in supporting. Other cells give off fibers which branch repeatedly. Some of the fibers start from the epithelial cells lining the ventricles of the brain and central canal of the medulla spinalis, and pass through nervous tissue, branching repeatedly to end in slight enlargements on the pia mater. Thus, neuroglia is evidently a connective tissue in function, but is not so in development. It is ectodermal in origin, whereas all connective tissues are mesodermal. Nerve cells. Nerve cells are largely aggregated in the gray substance of the brain and medulla spinalis, but smaller collections of these cells also form the swellings called ganglia seen on many nerves. These latter are found chiefly upon the spinal and cranial nerve roots and in connection with the sympathetic nerves. The nerve cells vary in shape and size and have one or more processes. They may be divided for purposes of description into three groups according to the number of processes which they possess. 1. Unipolar cells, which are found in the spinal ganglia. The single process after a short course divides in a T-shaped manner. 2. Bipolar cells, also found in the spinal ganglia, when the cells are in an embryonic condition. They are best demonstrated in the spinal ganglia of fish. Sometimes the processes come off from opposite poles of the cell, and the cell then assumes a spindle shape. In others, both processes emerge at the same point. In some cases, where two fibers are apparently connected with a cell, one of the fibers is really derived from an adjoining nerve cell and is passing to end in a ramification around the ganglion cell. Or, again, it may be coiled spirally around a nerve process which is issuing from the cell. 3. Multipolar cells, which are pyramidal or stellate in shape and characterized by their large size and by the numerous processes which issue from them. The processes are of two kinds. One of them is termed the axis cylinder process, or axon, because it becomes the axis cylinder of a nerve fiber. The others are termed the protoplasmic processes, or dendrons. They begin to divide and subdivide as soon as they emerge from the cell, and finally end in minute twigs and become lost among the other elements of the nervous tissue. The body of the nerve cell, known as the cyton, consists of a finely fibrillated protoplasmic material of a reddish or yellowish-brown color, which occasionally presents patches of a deeper tint, caused by the aggregation of pigment granules at one side of the nucleus, as in the substantia nigra and locus coeruleus of the brain. The protoplasm also contains peculiar angular granules which stain deeply with basic dyes, such as methylene blue. These are known as Nissel's granules. They extend into the dendritic processes, but not into the axis cylinder. The small clear area at the point of exit of the axon is termed the cone of origin. These granules disappear, chromatolysis, during fatigue or after prolonged stimulation of the nerve fibers connected with the cells. They are supposed to represent a store of nervous energy, and in various mental diseases are deficient or absent. The nucleus is, as a rule, a large, well-defined spherical body, often presenting an intranuclear network and containing a well-marked nucleolus. In addition to the protoplasmic network described above, each nerve may be shown to have delicate neurofibrils running through its substance. These fibrils are continuous to the fibrils of the axon and are believed to convey nerve impulses. Golgi has also described an extracellular network, which is probably a supporting structure. Nerve fibers. Nerve fibers are found universally in the peripheral nerves and in the white substance of the brain and medulla spinalis. They are of two kinds, that is, medullated, or white fibers, and non-medullated, or gray fibers. The medullated fibers form the white part of the brain and medulla spinalis, and also the greater part of every cranial and spinal nerve, and give to these structures their opaque white aspect. When perfectly fresh, they appear to be homogeneous, but soon after removal from the body, each fiber presents, when examined by transmitted light, a double outline or contour, as if consisting of two parts. The central portion is named the axis cylinder. Around this is a sheath of fatty material, staining black with osmic acid, named the white substance of Schwann or medullary sheath, which gives to the fiber its double contour, and the whole is enclosed in a delicate membrane, the neurolemma, primitive sheath, or nucleated sheath of Schwann. The axis cylinder is the essential part of the nerve fiber and is always present. The medullary sheath and the neurolemma are occasionally absent, especially at the origin and termination of the nerve fiber. The axis cylinder undergoes no interruption from its origin in the nerve center to its peripheral termination, and must be regarded as a direct prolongation of a nerve cell. It constitutes about 1/2 or 1/3 of the nerve fiber, being greater in proportion in the fibers of the central organs than in those of the nerves. It is quite transparent, and is therefore indistinguishable in a perfectly fresh and natural state of the nerve. It is made up of exceedingly fine fibrils, which stain darkly with gold chloride, and at its terminations may be seen to break up into these fibrillae. The fibrillae have been termed the primitive fibrillae of Schulze. The axis cylinder is said by some to be enveloped in a special reticular sheath, which separates it from the medullary sheath and is composed of a substance called neurokeratin. The more common opinion is that this network or reticulum is contained in the white matter of Schwann, and by some it is believed to be produced by the action of the reagents employed to show it. The medullary sheath, or white matter of Schwann, is regarded as being fatty matter in a fluid state, which insulates and protects the essential part of the nerve, the axis cylinder. It varies in thickness, in some forming a layer of extreme thinness so as to be scarcely distinguishable, in others forming about 1/2 of the nerve fiber. The variation in diameter of the nerve fibers, from 2 to 16 microns, depends mainly upon the amount of white substance, though the axis cylinder also varies within certain limits. The medullary sheath undergoes interruptions in its continuity at regular intervals, giving the fiber the appearance of constriction at these points. These are known as the nodes of Ranvier. The portion of nerve fiber between two nodes is called an internodal segment. The neurolemma, or primitive sheath, is not interrupted at the nodes, but passes over them as a continuous membrane. If the fiber be treated with silver nitrate, the reagent penetrates the neurolemma at the nodes, and on exposure to light reduction takes place, giving rise to the appearance of black crosses, Ranvier's crosses, on the axis cylinder. There may also be seen transverse lines beyond the nodes termed Frohman's lines. The significance of these is not understood. In addition to these interruptions, oblique clefts may be seen in the medullary sheath, subdividing it into irregular portions which are termed medullary segments, or segments of Lantermann. There is reason to believe that these clefts are artificially produced in the preparation of these specimens. Medullated nerve fibers, when examined in the fresh condition, frequently present a beaded or varicose appearance. This is due to manipulation and pressure causing the oily matter to collect into drops, and in consequence of the extreme delicacy of the primitive sheath, even slight pressure will cause the transudation of the fatty matter, which collects as drops of oil outside the membrane. The neurolemma, or primitive sheath, presents the appearance of a delicate structureless membrane. Here and there beneath it, and situated in depression in the white matter of Schwann, are nuclei surrounded by a small amount of protoplasm. The nuclei are oval and somewhat flattened, and bear a definite relation to the nodes of Ranvier, one nucleus generally lying in the center of each internode. The primitive sheath is not present in all medullated nerve fibers, being absent in those fibers which are found in the brain and medulla spinalis. Wallerian degeneration. When nerve fibers are cut across, the central ends of the fibers degenerate as far as the first node of Ranvier, but the peripheral ends degenerate simultaneously throughout their whole length. The axons break up into fragments and become surrounded by drops of fatty substance which are formed from the breaking down of the medullary sheath. The nuclei of the primitive sheath proliferate, and finally absorption of the axons and fatty substance occurs. If the cut ends of the nerve be sutured together, regeneration of the nerve fibers takes place by the downgrowth of axons from the central end of the nerve. At one time it was believed that the regeneration was peripheral in origin, but this has been disproved. The proliferated nuclei in the peripheral portions taking part merely in the formation of the so-called scaffolding along which the new axons pass. Non-medullated fibers. Most of the fibers in the sympathetic nervous system and some of the cerebrospinal consist of gray or gelatinous nerve fibers, fibers of Ramac. Each of these contain an axis cylinder to which nuclei are applied at intervals. These nuclei are believed to be in connection with a delicate sheath corresponding with the neurolemma of the medullated nerve fiber. In external appearance, the non-medullated nerve fibers are semi-transparent and gray or yellowish-gray. The individual fibers vary in size, generally averaging about half the size of the medullated fibers. End of section 1. Section 2 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. Structure of the Nervous System, Part 2. Structure of the peripheral nerves and ganglia. The cerebrospinal nerves consist of numerous nerve fibers collected together and enclosed in membranous sheaths. A small bundle of fibers enclosed in a tubular sheath is called a funiculus. If the nerve is of small size, it may consist of only a single funiculus, but if large, the funiculi are collected together into larger bundles of fasciculi, which are bound together in a common membranous investment. In structure, the common membranous investment or sheath of the whole nerve, epineurium, as well as the septa given off from it to separate the fasciculi, consist of connective tissue composed of white and yellow elastic fibers, the latter existing in great abundance. The tubular sheath of the funiculi, perineurium, is a fine, smooth, transparent membrane which may be easily separated in the form of a tube from the fibers it encloses. In structure, it is made up of connective tissue, which has a distinctly lamellar arrangement. The nerve fibers are held together and supported within the funiculus by delicate connective tissue called the endoneurium. It is continuous with septae which pass inward from the innermost layer of the perineurium and shows a ground substance in which are embedded fine bundles of fibrous connective tissue running for the most part longitudinally. It serves to support capillary vessels arranged so as to form a network with elongated meshes. The cerebrospinal nerves consist almost exclusively of medullated nerve fibers, only a very small proportion of non-medullated being present. The blood vessels supplying a nerve end in a minute capillary plexus, the vessels composing which pierce the perineurium and run, for the most part, parallel with the fibers. They are connected together by short transverse vessels, forming narrow oblong meshes, similar to the capillary system of muscle. Fine, non-medullated nerve fibers, vasomotor fibers, accompany these capillary vessels, and break up into elementary fibrils, which form a network around the vessels. Horsley has demonstrated certain medullated fibers running in the epineurium and terminating in small spheroidal tactile corpuscles, or end bulbs of Krauss. These nerve fibers, which Marshall believes to be sensory, and which he has termed nervi nervorum, are considered by him to have an important bearing upon certain neuralgic pains. The nerve fibers, so far as it is present known, do not coalesce, but pursue an uninterrupted course from the center to the periphery. In separating a nerve, however, into its component funiculi, it may be seen that these do not pursue a perfectly insulated course, but occasionally join at very acute angle with other funiculi proceeding in the same direction. From this, branches are given off to join again in like manner with other funiculi. It must be distinctly understood, however, that in these communications the individual nerve fibers do not coalesce, but merely pass into the sheath of the adjacent nerve, become intermixed with nerve fibers, and again pass on to intermingle with the nerve fibers in some adjoining funiculus. Nerves, in their course, subdivide into branches, and these frequently communicate with branches of a neighboring nerve. The communications which thus take place form what is called a plexus. Sometimes a plexus is formed by the primary branches of the trunks of the nerves, as the cervical, brachial, lumbar, and sacral plexuses, and occasionally by the terminal funiculi, as in the plexus formed at the periphery of the body. In the formation of a plexus, the component nerves divide, then join, and again subdivide in such a complex manner that the individual funiculi become interlaced most intricately, so that each branch leaving a plexus may contain filaments from all the primary nerve trunks which form the plexus. In the formation also of smaller plexuses at the periphery of the body, there is a free interchange of the funiculi and primitive fibers. In each case, however, the individual fibers remain separate and distinct. It is probable that through this interchange of fibers, every branch passing off from a plexus has a more extensive connection with the spinal cord than if it had proceeded to its distribution without forming connections with other nerves. Consequently, the parts supplied by these nerves have more extended relations with the nervous centers. By this means, also, groups of muscles may be associated for combined action. The sympathetic nerves are constructed in the same manner as the cerebrospinal nerves, but consist mainly of non-medullated fibers, collected in funiculi and enclosed in sheaths of connective tissue. There is, however, in these nerves a certain admixture of medullated fibers. The number of these latter varies in different nerves and may be estimated by the color of the nerve. Those branches of the sympathetic which present a well-marked gray color are composed chiefly of non-medullated nerve fibers intermixed with a few medullated fibers, while those of a white color contain many of the latter fibers and few of the former. The cerebrospinal and sympathetic nerve fibers convey various impressions. The sensory nerves, called also centripetal or afferent nerves, transmit to the nervous centers impressions made upon the peripheral extremities of the nerves, and in this way the mind, through the medium of the brain, becomes conscious of external objects. The centrifugal or efferent nerves transmit impressions from the nervous centers to the parts to which the nerves are distributed. These impressions, either exciting muscular contraction or influencing the processes of nutrition, growth, and secretion. Origins and terminations of nerves. By the expression, quote, the termination of nerve fibers, end quote, is signified their connections with the nerve centers and with the parts they supply. The former are sometimes called their origins or central terminations, the latter their peripheral terminations. Origins of nerves. The origins in some cases is single, that is to say, the whole nerve emerges from the nervous center by a single root. In other instances, the nerve arises by two or more roots, which come off from different parts of the nerve center, sometimes widely apart from each other. And it often happens, when a nerve arises in this way by two roots, that the function of these two roots are different, as, for example, in the spinal nerves, each of which arises by two roots, the anterior of which is motor, and the posterior sensory. The point where the nerve root or roots emerge from the surface of the nervous center is named a superficial or apparent origin. But the fibers of the nerves can be traced for a certain distance into the substance of the nervous center to some portion of the gray matter, which constitutes the deep or real origin of the nerve. The centrifugal or efferent nerve fibers originate in the nerve cells of the gray substance, the axis cylinder processes of these cells being prolonged to form fibers. In the case of the centripetal or afferent nerves, the fibers grow inward either from the nerve cells in the origins of special sense, for example the retina, or from nerve cells in the ganglia. Having entered the nerve center, they branch and send their ultimate twigs among the cells, without, however, uniting with them. Peripheral terminations of nerves. Nerve fibers terminate peripherally in various ways, and these may be conveniently studied in the sensory and motor nerves respectively. The terminations of the sensory nerves are dealt with in the section on sense organs. Motor nerves can be traced into either unstriped or striped muscular fibers. In the unstriped or involuntary muscles, the nerves are derived from the sympathetic and are composed mainly of non-medullated fibers. Near their terminations, they divide into numerous branches which communicate and form intimate plexuses. At the junction of the branches, small triangular nuclear bodies, ganglion cells, are situated. From these plexuses, minute branches are given off which divide and break up into the ultimate fibrillae of which the nerves are composed. These fibrillae course between the involuntary muscle cells and, according to Elischer, terminate on the surfaces of the cells opposite the nuclei in minute swellings. In the striped or voluntary muscle, the nerves supplying the muscular fibers are derived from the cerebrospinal nerves and are composed mainly of medullated fibers. The nerve, after entering the sheath of the muscle, breaks up into fibers or bundles of fibers which form plexuses and gradually divide until, as a rule, a single nerve fiber enters a single muscular fiber. Sometimes, however, if the muscular fiber be long, more than one nerve fiber enters it. Within the muscular fiber, the nerve terminates in a special expansor called by Kuhn, who first accurately described it as a motor end plate. The nerve fiber, on approaching the muscular fiber, suddenly loses its medullary sheath. The neurolemma becomes continuous with the sarcolemma of the muscle, and only the axis cylinder enters the muscular fiber. There it at once spreads out, ramifying like the roots of a tree immediately beneath the sarcolemma, and becomes embedded in a layer of granular matter, containing a number of clear oblong nuclei, the whole constituting an end plate, from which the contractile wave of the muscular fiber is said to start. Ganglia are small aggregations of nerve cells. They are found on the posterior roots of the spinal nerve, on the sensory roots of the trigeminal, facial, glossopharyngeal, and vagus nerves, and on the acoustic nerves. They are also found in connection with the sympathetic nerves. On section, they are seen to consist of a reddish-gray substance traversed by numerous white fibers. They vary considerably in form and size. The largest are found in the cavity of the abdomen, the smallest, not visible to the naked eye, exist in considerable numbers upon the nerves distributed to the different viscera. Each ganglion is invested by a smooth and firm, closely adhering, membranous envelope consisting of dense areolar tissue. This sheath is continuous with the perineurium of the nerves and sends numerous processes into the interior to support the blood vessels supplying the substance of the ganglion. In structure, all ganglia are essentially similar, consisting of the same structural elements, that is, nerve cells and nerve fibers. Each nerve cell has a nucleated sheath which is continuous with the neurolemma of the nerve fiber with which the cell is connected. The nerve cells in the ganglia of the spinal nerves are piriform in shape and have each a single process. A short distance from the cell, and while still within the ganglion, this process divides in a T-shaped manner, one limb of the crossbar turning into the medulla spinalis, the other limb passing outward to the periphery. In the sympathetic ganglia, the nerve cells are multipolar and each has one axis cylinder process and several dendrons. The axon emerges from the ganglion as a non-medullated nerve fiber. Similar cells are found in the ganglia connected with the trigeminal nerve, and these ganglia are therefore regarded as the cranial portions of the sympathetic system. The sympathetic nervous system includes those portions of the nervous mechanism in which a medullated nerve fiber from the central nervous system passes to a ganglion, sympathetic or peripheral, from which fibers, usually non-medullated, are distributed to such structures, for instance blood vessels, as are not under voluntary control. The spinal and sympathetic ganglia differ somewhat in the size and disposition of the cells and in the number of nerve fibers entering and leaving them. In the spinal ganglia, the nerve cells are much larger and for the most part collected in groups near the periphery, while the fibers, which are mostly medullated, traverse the central portion of the ganglion. Whereas in the sympathetic ganglia, the cells are smaller and distributed in irregular groups throughout the whole ganglion. The fibers are also irregularly scattered. Some of the entering ones are medullated, while many of the leaving the ganglia are non-medullated. Neuron theory. The nerve cell and its processes collectively constitute what is termed a neuron. And Walden formulated the theory that the nervous system is built up of numerous neurons, quote, anatomically and genetically independent of one another, end quote. According to this theory, the neuron theory, the processes of one neuron only come into contact and are never in direct continuity with those of other neurons, while impulses are transmitted from one nerve cell to another through these points of contact, the synapses. The synapse or synaptic membranes seems to allow nervous impulses to pass in one direction only, namely from the terminals of the axis cylinder to the dendrons. This theory is based on the following facts. That is, 1. Embryonic nerve cells, or neuroblasts, are entirely distinct from one another. 2. When nervous tissues are stained by the Golgi method, no continuity is seen even between neighboring neurons. And 3. When degenerative changes occur in nervous tissue, either as the result of disease or experiment, they never spread from one neuron to another, but are limited to the individual neurons or groups of neurons primarily affected. It must, however, be added that within the past few years, the validity of the neuron theory had been called into question by certain eminent histologists, who maintain that by the employment of a more delicate histologic method, minute fibrils can be followed from one nerve cell to another. Their existence, however, in the living is open to question. Mott and Marinesco made careful examination of living cells using even the ultramicroscope and agree that neither Nissl bodies nor neurofibrils are present in the living state. For the present, we may look upon the neurons as the units or structural elements of the nervous system. All the neurons are present at birth, which are present in the adult. Their division ceases before birth. They are not all functionally active at birth, but gradually assume functional activity. There is no indication of any regeneration after the destruction of the cell body of any individual neurons. Fasciculi, tracts, or fiber systems, are groups of axons having homologous origin and homologous distribution, as regards their collaterals, subdivisions, and terminals, and are often named in accordance with their origin and termination, the name of the nucleus or the location of the cell body from which the axon or fiber arises, preceding that of the nucleus or location of its termination. A given topographical area seldom represents a pure tract, as in most cases fibers of different systems are mixed. End of section 2. Section 3 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. Development of the Nervous System, Part 1. Development of the nervous system. The entire nervous system is of ectodermal origin, and its first rudiment is seen in the neural groove, which extends along the dorsal aspect of the embryo. By the elevation and ultimate fusion of the neural folds, the groove is converted into the neural tube. The anterior end of the neural tube becomes expanded to form the 3 primary brain vesicles. The cavity of the tube is subsequently modified to form the ventricular cavities of the brain and the central canal of the medulla spinalis. From the wall of the nervous system elements and the neuroglia of the brain and medulla spinalis are developed. The medulla spinalis. At first, the wall of the neural tube is composed of a single layer of columnar ectodermal cells. Soon the side walls become thickened, while the dorsal and ventral parts remain thin and are named the roof and floor plates. A transverse section of the tube at this stage presents an oval outline, while its lumen has the appearance of a slit. The cells which constitute the wall of the tube proliferate rapidly, lose their cell boundaries, and form a syncytium. This syncytium consisted first of dense protoplasm with closely packed nuclei, but later it opens out and forms a looser meshwork, with the cellular strands arranged in a radiating manner from the central canal. 3 layers may now be defined: an internal or ependymal, an intermediate or mantle, and an external or marginal. The ependymal layer is ultimately converted into the ependyma of the central canal. The processes of its cells pass outward towards the periphery of the medulla spinalis. The marginal layer is devoid of nuclei and later forms a supporting framework for the white funiculi of the medulla spinalis. The mantle layer represents the whole of the future gray columns in the medulla spinalis. In it, the cells are differentiated into 2 sets, that is, spongioblasts, or young neuroglial cells, and germinal cells, which are the parents of the neuroblasts, or young nerve cells. The spongioblasts are at first connected to one another by filaments of the syncytium. In these, fibrils are developed so that as the neuroglial cells become defined, they exhibit their characteristic mature appearance with multiple processes extending from each cell. The germinal cells are large, round or oval, and first make their appearance between the ependymal cells on the sides of the central canal. They increase rapidly in number, so that by the 4th week they form an almost continuous layer on each side of the tube. No germinal cells are found in the roof or floor plates. The roof plate retains, in certain regions of the brain, its epithelial character. Elsewhere, its cells become spongioblasts. By subdivision, the germinal cells give rise to the neuroblast or young nerve cells, which migrate outward from the sides of the central canal into the mantle layer and neural crest, and at the same time become pear-shaped. The tapering part of the cell undergoes still further elongation and forms the axis cylinder of the cell. The lateral walls of the medulla spinalis continue to increase in thickness, and the canal widens out near its dorsal extremity and assumes a somewhat lozenge-shaped appearance. The widest part of the canal serves to subdivide the lateral wall of the neural tube into a dorsal or alar and a ventral or basal lamina, a subdivision which extends forward into the brain. At a later stage, the ventral part of the canal widens out, while the dorsal part is first reduced to a mere slit and then becomes obliterated by the approximation and fusion of its walls. The ventral part of the canal persists and forms the central canal of the adult medulla spinalis. The caudal end of the canal exhibits a conical expansion, which is known as the terminal ventricle. The ventral part of the mantle layer becomes thickened, and on cross-section appears as a triangular patch between the marginal and ependymal layers. This thickening is the rudiment of the anterior column of gray substance and contains many neuroblasts, the axis cylinders of which pass out through the marginal layer and form the anterior roots of the spinal nerves. The thickening of the mantle layer gradually extends in a dorsal direction and forms the posterior column of gray substance. The axons of many of the neuroblasts in the alar lamina run forward and cross in the floor plate to the opposite side of the medulla spinalis. These form the rudiment of the anterior white commissure. About the end of the 4th week, nerve fibers begin to appear in the marginal layer. The first to develop are the short intersegmental fibers from the neuroblast in the mantle zone, and the fibers of the dorsal nerve roots, which grow into the medulla spinalis from the cells of the spinal ganglia. By the 6th week, these dorsal root fibers form a well-defined oval bundle in the peripheral part of the alar lamina. This bundle gradually increases in size, and spreading towards the middle line, form the rudiment of the posterior funiculus. The long intersegmental fibers begin to appear about the 3rd month, and the cerebrospinal fibers about the 5th month. All nerve fibers are at first destitute of medullary sheaths. Different groups of fibers receive their sheaths at different times. The dorsal and ventral nerve roots about the 5th month, and the cerebrospinal fibers after the 9th month. by the growth of the anterior columns of gray substance, and by the increase in size of the anterior funiculi, a furrow is formed between the lateral halves of the cord anteriorly. This gradually deepens to form the anterior median fissure. The mode of formation of the posterior septum is somewhat uncertain. Many believe that it is produced by the growing together of the walls of the posterior part of the central canal, and by the development from its ependymal cells of a septum of fibrillated tissue which separates the future funicula gracilis. Up to the 3rd month of fetal life, the medulla spinalis occupies the entire length of the vertebral canal, and the spinal nerves pass outward at right angles to the medulla spinalis. From this time onward, the vertebral column grows more rapidly than the medulla spinalis, and the latter, being fixed above through its continuity with the brain, gradually assumes a higher position within the canal. By the 6th month, its lower end reaches only as far as the upper end of the sacrum. At birth, it is on a level with the 3rd lumbar vertebra, and in the adult with the lower border of the 1st or upper border of the 2nd lumbar vertebra. A delicate filament, the filum terminale, extends from its lower end as far as the coccyx. The spinal nerves. Each spinal nerve is attached to the medulla spinalis by an anterior or ventral and a posterior or dorsal root. The fibers of the anterior roots are formed by the axons of the neuroblast which lie in the ventral part of the mantle layer. These axons grow out through the overlying marginal layer and become grouped to form the anterior nerve root. The fibers of the posterior roots are developed from the cells of the spinal ganglia. Before the neural groove is closed to form the neural tube, a ridge of ectodermal cells, the ganglion ridge or neural crest, appears along the prominent margin of each neural fold. When the folds meet in the middle line, the two ganglion ridges fuse and form a wedge-shaped area along the line of closure of the tube. The cells of this area proliferate rapidly opposite the primitive segments and then migrate in a lateral and ventral direction to the sides of the neural tube, where they ultimately form a series of oval-shaped masses, the future spinal ganglia. These ganglia are arranged symmetrically on the two sides of the neural tube and, except in the region of the tail, are equal in number to the primitive segments. The cells of the ganglia, like the cells of the mantle layer, are of two kinds, that is, spongioblasts and neuroblasts. The spongioblasts develop into the neuroglial cells of the ganglia. The neuroblasts are at first round or oval in shape, but soon assume the form of spindles, the extremities of which gradually elongate into central and peripheral processes. The central processes grow medialward, and becoming connected with the neural tube, constitute the fibers of the posterior nerve roots, while the peripheral processes grow lateralwards to mingle with the fibers of the anterior root in the spinal nerve. As development proceeds, the original bipolar form of the cells changes. The two processes become approximated until they ultimately arise from a single stem in a T-shaped manner. Only in the ganglia of the acoustic nerve is the bipolar form retained. More recent observers hold, however, that the T form is derived from the branching of a single process which grows out from the cell. The anterior or ventral and the posterior or dorsal nerve roots join immediately beyond the spinal ganglion to form the spinal nerve, which then divides into anterior, posterior, and visceral divisions. The anterior and posterior divisions proceed directly to the areas of distribution without further association with ganglion cells. The visceral distributions are distributed to the thoracic, abdominal, and pelvic viscera, to reach which they pass through the sympathetic trunk, and many of the fibers form arborizations around the ganglion cells of this trunk. Visceral branches are not given off from all the spinal nerves. They form two groups, that is, thoracolumbar from the first or second thoracic to the second or third lumbar nerves, and pelvic from the second and third or third and fourth sacral nerves. The brain. The brain is developed from the anterior end of the neural tube, which at an early period becomes expanded into three vesicles, the primary cerebral vesicles. These are marked off from each other by intervening constrictions and are named the forebrain or prosencephalon, the midbrain or mesencephalon, and the hindbrain or rhombencephalon, the last being continuous with the medulla spinalis. As the result of unequal growth of these different parts, 3 flexures are formed, and the embryonic brain becomes bent on itself in a somewhat zigzag fashion. The 2 earliest flexures are concave ventrally and are associated with corresponding flexures of the whole head. The first flexure appears in the region of the midbrain and is named the ventral cephalic flexure. By means of it, the forebrain is bent in a ventral direction around the anterior end of the notochord and foregut, with the result that the floor of the forebrain comes to lie almost parallel with that of the hindbrain. This flexure causes the midbrain to become for a time the most prominent part of the brain, since its dorsal surface corresponds with the convexity of the curve. The second bend appears at the junction of the hindbrain and medulla spinalis. This is termed the cervical flexure, and increases from the 3rd to the end of the 5th week, when the hindbrain forms nearly a right angle with the medulla spinalis. After the 5th week, erection of the head takes place, and the cervical flexure diminishes and disappears. The third bend is named the pontine flexure, because it is found in the region of the future pons varioli. It differs from the other 2 in that A, its convexity is forward, and B, it does not affect the head. The lateral walls of the brain tube, like those of the medulla spinalis, are divided by internal furrows into alar, or dorsal and basal, or ventral laminae. The hindbrain, or rhombencephalon. The cavity of the hindbrain becomes the 4th ventricle. At the time when the ventral cephalic flexure makes its appearance, the length in the hindbrain exceeds the combined length of the other 2 vesicles. Immediately behind the midbrain, it exhibits a marked constriction, the isthmus rhombencephali, which is best seen when the brain is viewed from the dorsal aspect. From the isthmus, the anterior medullary velum and the superior peduncle of the cerebellum are formed. It is customary to divide the rest of the hindbrain into 2 parts, that is, an upper called the metencephalon, and a lower, the myelencephalon. The cerebellum is developed by a thickening of the roof, and the pons by a thickening of the floor and lateral walls of the metencephalon. The floor and lateral walls of the myelencephalon are thickened to form the medulla oblongata. Its roof remains thin and, retaining to a great extent its epithelial nature, is expanded in a lateral direction. Later, by the growth and backward extension of the cerebellum, the roof is folded inward towards the cavity of the 4th ventricle. It assists in completing the dorsal wall of this cavity and is also invaginated to form the ependymal covering of its choroid plexuses. Above it is continuous with the posterior medullary velum, below with the obex and lingulae. The development of the medulla oblongata resembles that of the medulla spinalis, but at the same time exhibits 1 or 2 interesting modifications. On transverse section, the myelencephalon at an early stage is seen to consist of 2 lateral walls connected across the midline by floor and roof plates. Each lateral wall consists of an alar and a basal lamina, separated by an internal furrow, the remains of which are represented in the adult brain by the sulcus limitans and the rhomboid fossa. The contained cavity is more or less triangular in outline, the base being formed by the roof plate, which is thin and greatly expanded transversely. Pear-shaped neuroblasts are developed in the alar and basal lamina, and their narrow stalks are elongated to form the axis cylinder of the nerve fibers. Opposite the furrow, or boundary between the alar and basal lamina, a bundle of nerve fibers attaches itself to the outer surface of the alar lamina. This is named the tractus solitarius, and is formed by the sensory fibers of the glossopharyngeal and vagus nerves. It is the homologue of the oval bundle seen in the medulla spinalis, and, like it, is developed by an ingrowth of fibers from the ganglia of the neural crest. At first, it is applied to the outer surface of the alar lamina, but it soon becomes buried owing to the growth over it of the neighboring parts. By the 5th week, the dorsal part of the alar lamina bends in a lateral direction along its entire length to form what is termed the rhombic lip. Within a few days, this lip becomes applied to and unites with the outer surface of the main part of the alar lamina, and so covers in the tractus solitarius and also the spinal root of the trigeminal nerve. The nodulus and flocculus of the cerebellum are developed from the rhombic lip. Neuroblasts accumulate in the mantle layer. Those in the basal lamina correspond with the cells in the anterior gray column of the medulla spinalis, and like them, give origin to motor nerve fibers. In the medulla oblongata they are, however, arranged in groups or nuclei instead of forming a continuous column. From the alar lamina and its rhombic lip, neuroblasts migrate into the basal lamina and become aggregated to form the olivary nuclei, while many send their axis cylinders through the floor plate to the opposite side and thus constitute the rudiment of the raphe of the medulla oblongata. By means of this thickening of the ventral portion, the motor nuclei are buried deeply in the interior and, in the adult, are found close to the rhomboid fossa. This is still further accentuated, A, by the development of the pyramids, which are formed about the 4th month by the downward growth of the motor fibrils from the cerebral cortex, and B, by the fibers which pass to and from the cerebellum. On the rhomboid fossa, a series of 6 temporary furrows appear. These are termed the rhombic grooves. They bear a definite relationship to certain of the cranial nerves. Thus, from before backward, the first and second grooves overlie the nucleus of the trigeminal, the third the nucleus of the facial, the fourth that of the abducent, the fifth that of the glossopharyngeal, and the sixth that of the vagus. The pons is developed from the ventrolateral wall of the metencephalon by a process similar to that which has been described for the medulla oblongata. The cerebellum is developed in the roof of the anterior part of the hindbrain. The alar lamina of this region become thickened to form 2 lateral plates, which soon fuse in the middle line and produce a thick lamina which roofs the upper part of the cavity of the hindbrain vesicle. This constitutes the rudiment of the cerebellum, the outer surface of which is originally smooth and convex. The fissures of the cerebellum appear first in the vermis and floccular region, and traces of them are found during the 3rd month. The fissures on the cerebellar hemispheres do not appear until the 5th month. The primitive fissures are not developed in the order of their relative size in the adult. Thus, the horizontal sulcus in the 5th month is merely a shallow groove. The best marked of the early fissures are A, the fissura prima between the developing culmen and diclive, and B, the fissura secunda between the future pyramid and uvula. The flocculus and nodule develop from the rhombic lip and are therefore recognizable as separate portions before any of the other cerebellar lobules. The groove produced by the bending over the rhombic lip is here known as the floccula fissure. When the 2 lateral walls fuse, the right and left floccular fissures join in the middle line, and their central part becomes the post-nodular fissure. On the ventricular surface of the cerebellar lamina, a transverse furrow, the incisura fastigii, appears and deepens to form the tent-like recess of the roof of the 4th ventricle. The rudiment of the cerebellum at first projects in a dorsal direction, but by the backward growth of the cerebrum, it is folded downward and somewhat flattened, and the thin roof plate of the 4th ventricle, originally continuous with the posterior border of the cerebellum, is projected inward towards the cavity of the ventricle. End of section 3. Section 4 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. Development of the Nervous System Part 2. The midbrain, or mesencephalon. The midbrain exists for a time as a thin-walled cavity of some size, and is separated from the isthmus rhombencephali behind and from the forebrain in front by slight constrictions. Its cavity becomes relatively reduced in diameter and forms the cerebral aqueduct of the adult brain. Its basal laminae increase in thickness to form the cerebral peduncles, which are at first of small size, but rapidly enlarge after the 4th month. The neuroblasts of these laminae are grouped in relation to the sides and floor of the cerebral aqueduct, and constitute the nuclei of the oculomotor and trochlear nerves, and of the mesencephalic root of the trigeminal nerve. By a similar thickening process, its alar laminae are developed into the quadrigeminal lamina. The dorsal part of the wall for a time undergoes expansion and presents an internal median furrow and a corresponding external ridge. These, however, disappear and the latter is replaced by a groove. Subsequently, two oblique furrows extend medialward and backward, and the thickened lamina is thus subdivided into the superior and inferior caliculi. The forebrain, or prosencephalon. A transverse section of the early forebrain shows the same parts as are displayed in similar sections of the medulla spinalis and medulla oblongata. That is, a pair of thick lateral walls connected by thin floor and roof plates. Moreover, each lateral wall exhibits a division into dorsal or alar and a ventral or basal lamina separated internally by a furrow termed the sulcus of Monro. This sulcus ends anteriorly at the medial end of the optic stalk, and in the adult brain is retained as a slight groove extending backward from the interventricular foramen to the cerebral aqueduct. At a very early period, in some animals before the closure of the cranial part of the neural tube, two lateral diverticula, the optic vesicles, appear, one on either side of the forebrain. For a time, they communicate with the cavity of the forebrain by relatively wide openings. The peripheral parts of the vesicles expand, while the proximal parts are reduced to tubular stalks, the optic stalks. The optic vesicles give rise to the retina and the epithelium on the back of the ciliary body and iris. The optic stalk is invaded by nerve fibers to form the optic nerve. The forebrain then grows forward, and from the alar lamina of this front portion, the cerebral hemispheres originate as diverticula, which rapidly expand to form two large pouches, one on either side. The cavities of these diverticula are rudiments of the lateral ventricles. They communicate with the median part of the forebrain cavity by relatively wide openings, which ultimately form the interventricular foramen. The median portion of the wall of the forebrain vesicle consists of a thin lamina, the lamina terminalis, which stretches from the interventricular foramen to the recess at the base of the optic stalk. The anterior part of the forebrain, including the rudiments of the cerebral hemispheres, is named a telencephalon, and its posterior portion is termed the diencephalon. Both of these contribute to the formation of the third ventricle. The diencephalon. From the alar lamina of the diencephalon, the thalamus, metathalamus, and epithalamus are developed. The thalamus arises as a thickening which involves the anterior two-thirds of the alar lamina. The two thalami are visible for a time on the surface of the brain, but are subsequently hidden by the cerebral hemispheres which grow backward over them. The thalami extend medialward and gradually narrow the cavity between them into a slit-like aperture, which forms the greater part of the third ventricle. Their medial surfaces ultimately adhere in part to each other, and their intermediate mass of the ventricle is developed across the area of contact. The metathalamus comprises the geniculate bodies, which originate as slight outward bulgings of the alar lamina. In the adult, the lateral geniculate body appears as an eminence on the lateral part of the posterior end of the thalamus, while the medial is situated on the lateral aspect of the midbrain. The epithalamus includes the pineal body, the posterior commissure, and the trigone habenula. The pineal body arises as an upward evagination of root plate immediately in front of the midbrain. This evagination becomes solid with the exception of its proximal part, which persists as the recessus pinealis. In lizards, the pineal evagination is elongated into a stalk, and its peripheral extremity is expanded into a vesicle in which a rudimentary lens and retina are formed. The stalk becomes solid and nerve fibers make their appearance in it, so that in these animals the pineal body forms a rudimentary eye. The posterior commissure is formed by the ingrowth of fibers into the depression behind and below the pineal evagination, and the trigonum habenulae is developed in front of the pineal recess. From the basal laminae of the diencephalon, the pars mammillaris hypothalami is developed. This comprises the corpora mammillaria and the posterior part of the tuber cinerum. The corpora mammillaria arise as a single thickening, which comes divided into two by a median furrow during the third month. The roof plate of the diencephalon, in front of the pineal body, remains thin and epithelial in character, and is subsequently invaginated by the choroid plexus of the third ventricle. The telencephalon. This consists of a median portion and two lateral diverticula. The median portion forms the anterior part of the cavity of the third ventricle, and is closed below and in front by the lamina terminalis. The lateral diverticula consist of outward pouchings of the alar laminae. The cavities represent the lateral ventricles, and their walls become thickened to form the nervous matter of the cerebral hemispheres. The roof plate of the telencephalon remains thin and is continuous in front with the lamina terminalis and behind with the roof plate of the diencephalon. In the basal lamina and the floor plate, the pars optica hypothalami is developed. This comprises the anterior part of the tuber cinerum, the infundibulum, and the posterior lobe of the hypophysis and the optic chiasma. The anterior part of the tuber cinerum is developed from the posterior part of the floor of the telencephalon. The infundibulum and posterior lobe of the hypophysis arises as a downward diverticulum from the floor. The most dependent part of the diverticulum becomes solid and forms the posterior lobe of the hypophysis. The anterior lobe of the hypophysis is developed from a diverticulum of the ectodermal lining of the stomadeum. The optic chiasma is formed by the meeting and partial decussation of the optic nerves, which subsequently grow backward as the optic tracts and end in the diencephalon. The cerebral hemispheres arise as diverticula of the alar lamina of the telencephalon. They increase rapidly in size and ultimately overlap the structures developed from the mid and hindbrains. This great expansion of the hemispheres is a characteristic feature of the brains of mammals and attains its maximum development in the brain of man. Elliott Smith divides each cerebral hemisphere into three fundamental parts, that is, the rhinencephalon, the corpus striatum, and the neopallium. The rhinencephalon represents the oldest part of the telencephalon and forms almost the whole of the hemisphere in fishes, amphibians, and reptiles. In man, it is feebly developed in comparison with the rest of the hemisphere, and comprises the following parts. That is, the olfactory lobe, consisting of the olfactory tract and bulb and trigonum olfactorium, the anterior perforated substance, the septum pellucidum, the subcallosal, supracallosal, and dentate gyri, the fornix, the hippocampus, and the uncus. The rhinencephalon appears as longitudinal elevation with a corresponding internal furrow on the undersurface of the hemisphere close to the lamina terminalis. It is separated from the lateral surface of the hemisphere by a furrow, the external rhinal fissure, and is continuous behind with that part of the hemisphere which will ultimately form the anterior end of the temporal lobe. The elevation becomes divided by a groove into an anterior and a posterior part. The anterior grows forward as a hollow stalk, the lumen of which is continuous with the anterior part of the ventricular cavity. During the third month, the stalk becomes solid and forms the rudiment of the olfactory bulb and tract. A strand of gelatinous tissue in the interior of the bulb indicates the position of the original cavity. From the posterior part, the anterior perforated substance and the piriform lobe are developed. At the beginning of the fourth month, the latter forms a curved elevation continuous behind with the medial surface of the temporal lobe and consisting from before backwards of the gyrus olfactorius lateralis, gyrus ambiens, and gyrus semilunaris, parts which in the adult brain are represented by the lateral root of the olfactory tract and the uncus. The position and connections of the remaining portions of the rhinencephalon are described with the anatomy of the brain. The corpus striatum appears in the fourth week as a triangular thickening of the floor of the telencephalon between the optic recess and the interventricular foramen, and continues behind with the thalamic part of the diencephalon. It increases in size, and by the second month is seen as a swelling in the floor of the furtio lateral ventricle. This swelling reaches as far as the posterior end of the primitive hemisphere, and when this part of the hemisphere grows backward and downward to form the temporal lobe, the posterior part of the corpus striatum is carried into the roof of the inferior horn of the ventricle, where it is seen as a tail of the caudate nucleus in the adult brain. During the 4th and 5th month, the corpus striatum becomes incompletely subdivided by the fibers of the internal capsule into two masses, an inner, the caudate nucleus, and an outer, the lentiform nucleus. In front, the corpus striatum is continuous with the anterior perforated substance. Laterally, it is confluent for a time with that portion of the wall of the vesicle which is developed into the insula, but this continuity is subsequently interrupted by the fibers of the external capsule. The neopallium forms the remaining and by far the greater part of the cerebral hemisphere. It consists as an early stage of a relatively large, more or less hemispherical cavity, the primitive lateral ventricle, enclosed by a thin wall from which the cortex of the hemisphere is developed. The vesicle expands in all directions, but more especially upward and backward, so that by the 3rd month, the hemispheres cover the diencephalon, by the 6th they overlap the midbrain, and by the 8th the hindbrain. The median lamina uniting the two hemispheres does not share in their expansion, and thus the hemispheres are separated by a deep cleft, the forerunner of the longitudinal fissure, and this cleft is occupied by a septum of mesodermal tissue which constitutes the primitive falx cerebri. Coincidentally with the expansion of the vesicle, its cavity is drawn out into 3 prolongations which represent the horn of the future lateral ventricle. The hinder end of the vesicle is carried downward and forward and forms the inferior horn. The posterior horn is produced somewhat later and associated with the backward growth of the occipital lobe of the hemisphere. The roof plate of the primitive forebrain remains thin and of an epithelial character. It is invaginated into the lateral ventricle along the median wall of the hemisphere. This invagination constitutes the choroidal fissure and extends from the interventricular foramen to the posterior end of the vesicle. Mesodermal tissue, continuous with that of the primitive falx cerebri and carrying blood vessels with it, spreads between the two layers of the invaginated fold and forms the rudiment of the tela choroidea. The margins of the tela become highly vascular and form the choroid plexuses, which for some months almost completely fill the ventricular cavities. The tela at the same time invaginates the epithelial roof of the diencephalon to form the choroid plexuses of the 3rd ventricle. By the downward and forward growth of the posterior end of the vesicle to form the temporal lobe, the choroidal fissure finally reaches from the interventricular foramen to the extremity of the inferior horn of the ventricle. Parallel with, but above and in front of the choroidal fissure, the median wall of the cerebral vesicle becomes folded outward and gives rise to the hippocampal fissure on the median surface and to a corresponding elevation, the hippocampus within the ventricular cavity. The gray or ganglionic covering of the wall of the vesicle ends at the inferior margin of the fissure, is a thickened edge. Beneath this, the marginal reticular layer, future white substance, is exposed, and its lower thinned edge is continuous, with the epithelial invagination covering the choroid plexus. As a result of the later downward and forward growth of the temporal lobe, the hippocampal fissure and the parts associated with it extend from the interventricular foramen to the end of the inferior horn of the ventricle. The thickened edge of gray substance becomes the gyrus dentatus, the fasciola cinerea, and the supra and subcallosal gyri, while the free edge of the white substance forms the fimbria hippocampi and the body and crus of the fornix. The corpus callosum is developed within the arch of the hippocampal fissure, and the upper part of the fissure forms in the adult brain the callosal fissure on the medial surface of the hemisphere. The commissures. The development of the posterior commissure has already been referred to. The gray commissures of the hemisphere, that is, the corpus callosum, the fornix, and the anterior commissures, arise from the lamina terminalis. About the 4th month, a small thickening appears in this lamina immediately in front of the interventricular foramen. The lower part of this thickening is soon constricted off, and fibers appear in it to form the anterior commissure. The upper part continues to grow with the hemispheres and is invaded by 2 sets of fibers. Transverse fibers, extending between the hemispheres, pass into its dorsal part, which is now differentiated as the corpus callosum. In rare cases, the corpus callosum is not developed. Into the ventral part, longitudinal fibers from the hippocampus pass to the lamina terminalis and through that structure to the corpora mammillaria. These fibers constitute the fornix. A small portion lying antero-inferiorly between the corpus callosum and the fornix is not invaded by the commissural fibers. It remains thin, and later a cavity, the cavity of the septum pellucidum, forms in its interior. Fissures and sulci. The outer surface of the cerebral hemispheres is at first smooth, but later it exhibits a number of elevations or convolutions, separated from each other by fissures and sulci, most of which make their appearance during the 6th or 7th month of fetal life. The term fissure is applied to such grooves as involve the entire thickness of the cerebral wall, and thus produce corresponding eminences in the ventricular cavity, while the sulci affect only the superficial part of the wall, and therefore leave no impression in the ventricle. The fissures comprise the choroidal and hippocampal already described, and two others, that is, the calcarine and collateral, which produce the swellings known respectively as the calcar avus and the collateral eminence in the ventricular cavity. Of the sulci, the following may be referred to, that is, the central sulcus, fissure of Rolando, which is developed in 2 parts, the intraparietal sulcus in 4 parts, and the cingulate sulcus in 2 or 3 parts. The lateral cerebral or sylvian fissure differs from all the other fissures in its mode of development. It appears about the 3rd month as a depression, the sylvian fossa, on the lateral surface of the hemisphere. This fossa corresponds with the position of the corpus striatum, and its floor is molded to form the insula. The intimate connection which exists between the cortex of the insula and the subjacent corpus striatum prevents this part of the hemisphere wall from expanding at the same rate as the portions which surround it. The neighboring parts of the hemisphere therefore gradually grow over and cover in the insula, and constitute the temporal, parietal, frontal, and orbital opercula of the adult brain. The frontal and orbital opercula are the last to form, but by the end of the 1st year after birth, the insula is completely submerged by the approximation of the opercula. The fissures separating the opposed margins of the opercula constitute the composite lateral cerebral fissure. If a section across the wall of the hemisphere about the 6th week be examined microscopically, it will be found to consist of a thin marginal or reticular layer, a thick ependymal layer, and a thin intervening mantle layer. Neuroblasts from the ependymal and mantle layers migrate into the deep part of the marginal layer and form the cells of the cerebral cortex. The nerve fibers, which form the underlying white substance of the hemispheres, consist at first of outgrowth from the cells of the corpora striata and the thalami. Later, the fibers from the cells of the cortex are added. Medullation of these fibers begins about the time of birth and is continuous until puberty. A summary of these parts, derived from the brain vesicles, is given in the following table. The hindbrain or rhombencephalon give rise to the myelencephalon, medencephalon, and isthmus rhombencephali. The myelencephalon gives rise to the medulla oblongata and lower part of the 4th ventricle, the medencephalon to the pons cerebellum and intermediate part of the 4th ventricle, and the isthmus rhombencephali, the anterior medullary velum, brachium conjunctiva cerebelli, and upper part of the 4th ventricle. The midbrain, or mesencephalon, gives rise to the cerebral peduncles, lamina quadrigemina, and cerebral aqueduct. The forebrain, or prosencephalon, gives rise to the diencephalon and telencephalon. The diencephalon gives rise to the thalamus, metathalamus, epithalamus, pars mammillaris hypothalami, and the posterior part of the 3rd ventricle. The telencephalon gives rise to the anterior part of the 3rd ventricle, pars optica hypothalami, cerebral hemisphere's lateral ventricles, and interventricular foramina. The cranial nerves. With the exception of the olfactory, optic, and acoustic nerves, which will be especially considered, the cranial nerves are developed in a similar manner to the spinal nerves. The sensory or afferent nerves are derived from the cells of the ganglion rudiments of the neural crest. The central processes of these cells grow into the brain and form the roots of the nerves, while the peripheral processes extend outward and constitute their fibers of distribution. It has been seen, in considering the development of the medulla oblongata, that the tractus solitarius, derived from the fibers which grow inward from the ganglion rudiments of the glossopharyngeal and vagus nerves, is the homologue of the oval bundle in the cord, which had its origin in the posterior nerve roots. The motor or efferent nerves arise as outgrowths of the neuroblast situated in the basal lamina of the mid and hindbrain. While, however, the spinal motor nerve roots arise in one series from the basal lamina, the cranial motor nerves are grouped into two sets, according as they spring from the medial or lateral parts of the basal lamina. To the former set belong the oculomotor, trochlear, abducent, and hypoglossal nerves. To the latter, the accessory and motor fibers of the trigeminal, facial, glossopharyngeal, vagus nerves. End of section 4. Section 5 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen, Anatomy of the Human Body, Part 4 by Henry Gray. The Spinal Cord, Part 1. The medulla spinalis, or spinal cord. The medulla spinalis, or spinal cord, forms the elongated, nearly cylindrical part of the central nervous system which occupies the upper two-thirds of the vertebral canal. Its average length in the male is about 45 centimeters, in the female from 42 to 43 centimeters, while its weight amounts to about 30 grams. It extends from the level of the upper border of the atlas to that of the lower border of the first or upper border of the second lumbar vertebra. Above, it is continuous with the brain. Below, it ends in a conical extremity, the conus medullaris, from the apex of which a delicate filament, the filum terminale, descends as far as the first segment of the coccyx. The position of the medulla spinalis varies with the movements of the vertebral column, its lower extremity being drawn slightly upward when the column is flexed. It also varies at different periods of life. Up to the third month of fetal life, the medulla spinalis is as long as the vertebral canal, but from this stage onward, the vertebral column elongates more rapidly than the medulla spinalis, so that by the end of the 5th month, the medulla spinalis terminates at the base of the sacrum, and at birth, about the 3rd lumbar vertebra. The medulla spinalis does not fill the part of the vertebral canal in which it lies. It is ensheathed by 3 protective membranes, separated from each other by 2 concentric spaces. The 3 membranes are named from without inward, the dura mater, the arachnoid, and the pia mater. The dura mater is a strong fibrous membrane which forms a wide tubular sheath. This sheath extends below the termination of the medulla spinalis and ends at a point cul-de-sac at the level of the lower border of the second sacral vertebra. The dura mater is separated from the wall of the vertebral canal by the epidural cavity, which contains a quantity of loose areolar tissue and a plexus of veins. Between the dura mater and the subjacent arachnoid is a capillary interval, the subdural cavity, which contains a small quantity of fluid, probably of the nature of lymph. The arachnoid is a thin, transparent sheath separated from the pia mater by a comparatively wide interval, the subarachnoid cavity, which is filled with cerebrospinal fluid. The pia mater closely invests the medulla spinalis and sends delicate septae into its substance. A narrow band, the ligamentum denticulatum, extends along each of the lateral surfaces and is attached by a series of pointed processes to the inner surfaces of the dura mater. 31 pairs of spinal nerves spring from medulla spinalis, each nerve having an anterior or ventral and a posterior or dorsal root, the latter being distinguished by the presence of an oval swelling, the spinal ganglion, which contains numerous nerve cells. Each root consists of several bundles of nerve fibers, and at its attachment extends for some distance along the side of the medulla spinalis. The pairs of spinal nerves are grouped as follows: cervical 8, thoracic 12, lumbar 5, sacral 5, coccygeal 1, and for convenience of description, the medulla spinalis is divided into cervical, thoracic, lumbar, and sacral regions, corresponding with the attachments of the different groups of nerves. Although no trace of transverse segmentation is visible on the surface of the medulla spinalis, it is convenient to regard it as being built up in a series of superimposed spinal segments, or neuromeres, each of which has a length equivalent to the extent of attachment of a pair of spinal nerves. Since the extent of attachment of the successive pairs of nerves varies in different parts, it follows that the spinal segments are of varying lengths. Thus, in the cervical region, they average about 13 millimeters, in the mid-thoracic region about 26 millimeters, while in the lumbar and sacral region they diminish rapidly from about 15 millimeters at the level of the first pair of lumbar nerves to about 4 millimeters opposite the attachment of the lower sacral nerves. As a consequence of the relevant inequality in the rates of growth of the medulla spinalis and vertebral column, the nerve roots, which in the early embryo pass transversely outward to reach their respective intervertebral foramina, become more and more oblique in direction from above downward, so that the lumbar and sacral nerves descend almost vertically to reach their points of exit. From the appearance these nerves present at the attachment to the medulla spinalis, and from their great length, they are collectively termed the cauda equina. The filum terminale is the delicate filament, about 20 centimeters in length, prolonged downward from the apex of the conus medullaris. It consists of two parts, an upper and a lower. The upper part, or filum terminale internum, measures about 15 centimeters in length and reaches as far as the lower border of the second sacral vertebra. It is contained within the tubular sheath of dura mater and is surrounded by the nerves forming the cauda equina, from which it can be readily recognized by its bluish-white color. The lower part, or filum terminale externum, is closely invested by and is adherent to the dura mater. It extends downward from the apex of the tubular sheath and is attached to the back of the first segment of the coccyx. The filum terminale consists mainly of fibrous tissue, continuous above with that of the pia mater. Adhering to its outer surface, however, are a few strands of nerve fibers which probably represent rudimentary second and third coccygeal nerves. Further, the central canal of the medulla spinalis extends downward into it for 5 or 6 centimeters. Enlargements. The medulla spinalis is not quite cylindrical, being slightly flattened from before backward. It also presents two swellings or enlargements, an upper or cervical and a lower or lumbar. The cervical enlargement is the more pronounced and corresponds with the attachment of the large nerves which supply the upper limbs. It extends from about the third cervical to the second thoracic vertebra, its maximal circumference about 38 millimeters being on a level with the attachment of the sixth pair of cervical nerves. The lumbar enlargement gives attachment to the nerves which supply the lower limbs. It commences about the level of the ninth thoracic vertebra and reaches its maximum circumference of about 33 millimeters opposite the last thoracic vertebra, below which it tapers rapidly into the conus medullaris. Fissures and sulci. An anterior median fissure and a posterior median sulcus incompletely divide the medulla spinalis into two symmetrical parts, which are joined across the midline by a commissural band of nervous matter. The anterior median fissure, fissura mediana anterior, has an average depth of about 3 millimeters, but this is increased in the lower part of the medulla spinalis. It contains a double fold of pia mater, and its floor is formed by a transverse band of white substance, the anterior white commissure, which is perforated by blood vessels on their way to or from the central part of the medulla spinalis. The posterior median sulcus, sulcus medianus posterior, is very shallow. From it, a septum of neuroglia reaches rather more than halfway into the substance of the medulla spinalis. This septum varies in depth from 4 to 6 millimeters, but diminishes considerably in the lower part of the medulla spinalis. On either side of the posterior median sulcus and at a short distance from it, the posterior nerve roots are attached along a vertical furrow named the posterior lateral sulcus. The portion of the medulla spinalis which lies between this and the posterior median sulcus is named the posterior funiculus. In the cervical and upper thoracic regions, this funiculus presents a longitudinal furrow, the posterior intermediate sulcus. This marks the position of a septum which extends into the posterior funiculus and subdivides it into two fasciculi, a medial, named the fasciculus gracilis, tract of Gall, and a lateral, the fasciculus cuneatus, tract of Burdach. The portion of the medulla spinalis which lies in front of the posterior lateral sulcus is termed the anterolateral region. The anterior nerve roots, unlike the posterior, are not attached in linear series, and their position of exit is not marked by a sulcus. They arise by separate bundles which spring from the anterior column of the gray substance and, passing forward through the white substance, emerge over an area of some slight width. The most lateral of these bundles is generally taken as a dividing line which separates the anterior lateral region into two parts, that is, an anterior funiculus between the anterior median fissure and the most lateral of the anterior nerve roots, and a lateral funiculus between the exit of these roots and the posterior lateral sulcus. In the upper part of the cervical region, a series of nerve roots passes outward through the lateral funiculus of the medulla spinalis. These unite to form the spinal portion of the accessory nerve, which runs upward and enters the cranial cavity through the foramen magnum. The internal structure of the medulla spinalis. On examining a transverse section of the medulla spinalis, it is seen to consist of gray and white nervous substance, the former being enclosed within the latter. Gray substance. Substantia grisea centralis. The gray substance consists of two symmetrical portions, one in each half of the medulla spinalis. These are joined across the middle line by a transverse commissure of gray substance, through which runs a minute canal, the central canal, just visible to the naked eye. In a transverse section, each half of the gray substance is shaped like a comma or crescent, the concavity of which is directed laterally, and these, together with the intervening gray commissure, present the appearance of the letter H. An imaginary coronal plane through the central canal serves to divide each crescent into an anterior or ventral and a posterior or dorsal column. The anterior column, columna anterior, anterior cornu, directed forward, is broad and of a rounded or quadrangular shape. Its posterior part is termed the base and its anterior part the head, but these are not differentiated from each other by any well-defined constriction. It is separated from the surface of the medulla spinalis by a layer of white substance which is traversed by the bundles of the anterior nerve roots. In the thoracic region, the posterior lateral part of the anterior column projects lateralwards as a triangular field, which is named the lateral column. Columna lateralis, the lateral cornu. The posterior column, columna posterior, posterior cornu, is long and slender and is directed backwards and lateralwards. It reaches almost as far as the posterior lateral sulcus, from which it is separated by a thin layer of white substance, the tract of Lissauer. It consists of a base, directly continuous with the base of the anterior horn, and a neck or slightly constricted portion, which is succeeded by an oval or fusiform area termed the head, of which the apex approaches the posterior lateral sulcus. The apex is capped by a V-shaped or crescentic mass of translucent gelatinous neuroglia, termed the substantia gelatinosa of Rolando, which contains both neuroglial cells and small nerve cells. Between the anterior and posterior columns, the gray substance extends as a series of processes into the lateral funiculus to form a network called the formatio reticularis. The quantity of gray substance, as well as the form which it presents on transverse section, varies markedly at different levels. In the thoracic region, it is small, not only in amount, but relatively to the surrounding white substance. In the cervical and lumbar enlargements, it is greatly increased. In the latter, and especially in the conus medullaris, its proportion to the white substance is greatest. In the cervical region, its posterior column is comparatively narrow, while its anterior is broad and expanded. In the thoracic region, both columns are attenuated, and the lateral column is evident. In the conus medullaris, the gray substance assumes the form of two oval masses, one in each half of the cord, connected together by a broad gray commissure. The central canal, canalus centralis, runs throughout the entire length of medulla spinalis. The portion of gray substance in front of the canal is named the anterior gray commissure, that behind it, the posterior gray commissure. The former is thin and is in contact anteriorly with the anterior white commissure. It contains a couple of longitudinal veins, one on either side of the middle line. The posterior gray commissure reaches from the central canal to the posterior median septum and is thinnest in the thoracic region and thickest in the conus medullaris. The central canal is continued upward through the lower part of the medulla oblongata and opens into the fourth ventricle of the brain. Below, it reaches for a short distance into the filum terminale. In the lower part of the conus medullaris, it exhibits a fusiform dilatation, the terminal ventricle. This has a vertical measurement of from 8 to 10 millimeters, is triangular on cross-section, with its base directed forward, and tends to undergo obliteration after the age of 40 years. Throughout the cervical and thoracic regions, the central canal is situated in the anterior third of the medulla spinalis. In the lumbar enlargement, it is near the middle, and in the conus medullaris, it approaches the posterior surface. It is filled with cerebrospinal fluid and lined by ciliated columnar epithelium, outside of which is an encircling band of gelatinous substance, the substantia gelatinosa centralis. This gelatinous substance consists mainly of neuroglia, but contains a few nerve cells and fibers. It is traversed by processes from the deep ends of the columnar ciliated cells which line the central canal. End of section 5. Section number 6 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Spinal Cord Part 2. Structure of the gray substance. The gray substance consists of numerous nerve cells and nerve fibers held together by neuroglia. Throughout the greater part of the gray substance, the neuroglia presents the appearance of a sponge-like network, but around the central canal and on the apices of the posterior columns, it consists of the gelatinous substance already referred to. The nerve cells are multipolar and vary greatly in size and shape. They consist of 1. Motor cells of large size, which are situated in the anterior horn, and are especially numerous in the cervical and lumbar enlargements. The axons of most of these cells pass out from the anterior nerve roots, but before leaving the white substance, they frequently give off collaterals, which re-enter and ramify in the gray substance. 2. Cells of small or medium size, whose axons pass into the white matter, where some pursue an ascending and others a descending course, but most of them divide in a T-shaped manner into descending and ascending processes. They give off collaterals which enter and ramify into the gray substance, and the terminations of the axons behave in a similar manner. The lengths of these axons vary greatly. Some are short and pass only between the joining spinal segments, while others are longer and connect more distant segments. These cells and their processes constitute a series of association or intersegmental neurons, which link together the different parts of the medulla spinalis. The axons of most of these cells are confined to the side of the medulla spinalis in which the nerve cells are situated, but some cross to the opposite side through the anterior commissures and are termed crossed commissural fibers. Some of these latter end directly in the gray substance, while others enter the white substance and ascend or descend in it for varying distances, before finally terminating in the gray substance. 3. Cells of the type 2 of Golgi, limited for the most part to the posterior column, are found also in the substantia gelatinosa of Rolando. Their axons are short and entirely confined to the gray substance, in which they break up into numerous fine filaments. Most of the nerve cells are arranged in longitudinal columns and appear as groups on transverse section. Nerve cells in the anterior column. The nerve cells in the anterior column are arranged in columns of varying length. The longest occupies the medial part of the anterior column and is named the anteromedial column. It is well marked in C4, C5, again from C8 to L4. It disappears in L5 and S1, but is well marked in S2, S3, and S4. Open parens, Bruce, close parens, see footnote. Footnote, Topical Atlas of the Spinal Cord, 1901. End footnote. Behind it is a dorsomedial column of small cells, which is not represented in L5, S1, S2, nor below S4. Its axons probably pass into the dorsal rami of the spinal nerves to supply the dorsal musculature of the spinal column. In the cervical and lumbar enlargements, where the anterior column is expanded in a lateral direction, the following additional columns are present, that is, A. Anterolateral, which consists of two groups, one in C4, C5, C6, the other in C6, C7, C8 in the cervical enlargement, and of a group from L2 to S2 in the lumbosacral enlargement. B. Posterior lateral in the lower 5 cervical, lower 4 lumbar, and upper 3 sacral segments. C. Posterior lateral in the last cervical, first thoracic, and upper 3 sacral segments. And D. Central in the lower 4 lumbar and upper 2 sacral segments. These cell groups are evidently related to the nerve roots of the brachial and sacral plexuses and supply fibers to the muscles of the arm and leg. Throughout the base of the anterior column are scattered solitary cells, the axons of some of which form cross-commissural fibers, while others constitute the motor fibers of the posterior nerve roots. Footnote: Lenhasek and Cahal found that in the chick embryo, the axons of a few of these nerve cells passed backwards through the posterior column and emerged as the motor fibers of the posterior nerve roots. These fibers are said to control the peristaltic movement of the intestine. Their presence in man has not yet been determined. End footnote. Nerve cells in the lateral column. These form a column which is best marked where the lateral gray column is differentiated, that is, in the thoracic region, but it can be traced throughout the entire length of the medulla spinalis in the form of groups of small cells which are situated in the anterior part of the formatio reticularis. In the upper part of the cervical region and lower part of the medulla oblongata, as well as in the 3rd and 4th sacral segments, this column is again differentiated. In the medulla, it is known as the lateral nucleus. The cells of this column are fusiform or star-shaped and of a medium size. The axons of some of them pass into the anterior nerve roots by which they are carried to the sympathetic nerve. They constitute the white rami and are sympathetic or visceral efferent fibers. They are also known as preganglionic fibers of the sympathetic system. The axons of others pass into the anterior and lateral funiculi, where they become longitudinal. Nerve cells in the posterior column. 1. The dorsal nucleus, nucleus dorsalis, column of Clark, occupies the medial part of the base of the posterior column and appears on the transverse section as a well-defined oval area. It begins below at the level of the 2nd or 3rd lumbar nerve and reaches its maximum size opposite the 12th thoracic nerve. Above the level of the 9th thoracic nerve, its size diminishes and the column ends opposite the last cervical or 1st thoracic nerve. It represented, however, in the other regions by scattered cells which become aggregated to form a cervical nucleus opposite the 3rd cervical nerve and a sacral nucleus in the middle and lower part of the sacral region. Its cells are of medium size and of an oval or piriform shape. Their axons pass into the peripheral part of the lateral funiculus of the same side and there ascend, probably in the dorsospinocerebellar, direct cerebellar, fasciculus. 2. The nerve cells in the substantia gelatinosa of Rolando are arranged in 3 zones: a posterior or marginal of large angular or fusiform cells, an intermediate of small fusiform cells, and an anterior of star-shaped cells. The axons of these cells pass into the lateral and posterior funiculi and there assume a vertical course. In the anterior zone, some Golgi cells are found whose short axons ramify in the gray substance. 3. Solitary cells of varying forms and size are scattered throughout the posterior column. Some of these are grouped to form the posterior basal column in the base of the posterior column, lateral to the dorsal nucleus. The posterior basal column is well marked in the gorilla, Waldeyer, but is ill-defined in man. The axons of its cells pass partly to the posterior and lateral funiculi of the same side, and partly through the anterior white commissure to the lateral funiculus of the opposite side. Golgi cells, type 2, located in this region, send axons to the lateral and ventral columns. A few star-shaped or fusiform nerve cells of varying size are found in the substantia gelatinosa centralis. Their axons pass into the lateral funiculus of the same or of the opposite side. The nerve fibers in the gray substance form a dense interlacement of minute fibrils among the nerve cells. This interlacement is formed partly of axons which pass from the cells in the gray substance to enter the white funiculi or nerve roots, partly of the axons of Golgi cells, which ramify only in the gray substance, and partly of collaterals from the nerve fibers in the white funiculi which, as already stated, enter the gray substance and ramify within it. White substance, substantia alba. The white substance of the medulla spinalis consists of medullated nerve fibers embedded in a sponge-like network of neuroglia, and is arranged in 3 funiculi, anterior, lateral, and posterior. The anterior funiculus lies between the anterior median fissure and the most lateral of the anterior nerve roots, the lateral funiculus between these nerve roots and the posterior lateral sulcus, and the posterior funiculus between the posterior lateral and the posterior median sulci. The fibers vary greatly in thickness, the smaller being found in the fasciculus gracilis, the tract of Lissauer, and inner part of the lateral funiculus, while the largest are situated in the anterior funiculus and in the peripheral part of the lateral funiculus. Some of the nerve fibers assume a more or less transverse direction, as, for example, those which cross from side to side in the anterior white commissure. But the majority pursue a longitudinal course and are divisible into 1. Those connecting the medulla spinalis with the brain and conveying impulses to or from the latter, and 2. Those which are confined to the medulla spinalis and link together its different segments, that is, intersegmental or association fibers. Nerve fasciculi. The longitudinal fibers are grouped into more or less definite bundles or fasciculi. These are not recognizable from each other in the normal state, and their existence has been determined by the following methods. 1. A. Waller discovered that if a bundle of nerve fibers be cut, the portions of the fibers which are separated from their cells rapidly degenerate and become atrophied, while the cells and the parts of the fibers connected with them undergo little alteration. See footnote. Somewhat later, a change, termed chromatolysis, takes place in the nerve cells, and consists of a breaking down and an ultimate disappearance of the Nissl bodies. Further, the body of the cell is swollen, the nucleus displaced towards the periphery, and the part of the axon still attached to the altered cell is diminished in size and somewhat atrophied. Under favorable conditions, the cell is capable of reassuming its normal appearance, and its axon may grow again. End footnote. This is known as Wallerian degeneration. Similarly, if a group of nerve cells be destroyed, the fibers arising from them undergo degeneration. Thus, if the motor cells of the cerebral cortex be destroyed, or if the fibers arising from these cells be severed, a descending degeneration from the seat of injury takes place in the fibers. In the same manner, if a spinal ganglion be destroyed, or the fibers which pass from it into the medulla spinalis be cut, an ascending degeneration will extend along these fibers. 2. Pathological changes, especially in man, have given important information by causing ascending and descending degeneration. 3. By tracing the development of the nervous system, it has been observed that at first the nerve fibers are merely naked axis cylinders, and that they do not all acquire their medullary sheaths at the same time. Hence the fibers can be grouped into different bundles according to the dates at which they receive their medullary sheaths. 4. Various methods of staining nervous tissue are of great value in tracing the course and mode of termination of the axis cylinder processes. Fasciculi in the anterior funiculus. Descending fasciculi. The anterior cerebrospinal, fasciculus cerebrospinalis anterior, direct pyramidal tract, which is usually small but varies inversely in size with the lateral cerebrospinal funiculus. It lies close to the anterior median fissure and is present only in the upper part of the medulla spinalis, gradually diminishing in size as it descends. It ends about the middle of the thoracic region. It consists of descending fibers which arise from cells in the motor area of the cerebral hemisphere of the same side, and which, as they run downward in the medulla spinalis, cross in succession through the anterior white commissure to the opposite side, where they end, either directly or indirectly, by arborizing around the motor cells in the anterior column. A few of its fibers are said to pass to lateral columns of the same side and to the gray matter at the base of the posterior column. They conduct voluntary motor impulses from the precentral gyrus to the motor centers of the cord. The vestibulospinal fasciculus, situated chiefly in the marginal part of the funiculus and mainly derived from the cells of Dieter's nucleus, of the same and opposite side, that is, the chief terminal nucleus of the vestibular nerve. Fibers are also contributed to this fasciculus from scattered cells of the articular formation of the medulla oblongata, the pons, and the midbrain, tegmentum. The other terminal nuclei of the vestibular nerve also contributes fibers. In the brainstem, these fibers form part of the median longitudinal bundle. The fasciculus can be traced to the sacral region. Its terminals and collaterals end either directly or indirectly among the motor cells of the anterior column. This fasciculus is probably concerned with equilibratory reflexes. The tectospinal fasciculus, situated partly in the anterior and partly in the lateral funiculus, is mainly derived from the opposite superior colliculus of the midbrain. The fibers from the superior colliculus cross the median raphe and the fountain decussation of Meinert and descend as the ventral longitudinal bundle in the reticular formation of the brainstem. Its collaterals and terminals end either directly or indirectly among the motor cells of the anterior column of the same side. Since the superior colliculus is an important visual reflex center, the tectospinal fasciculus is probably concerned with visual reflexes. Ascending fasciculi. The ventral spinothalamic fasciculus, situated in the marginal part of the funiculus and intermingled more or less with the vestibulospinal fasciculus, is derived from cells in the posterior column or intermediate gray matter of the opposite side. Their axons cross in the anterior commissure. This is a somewhat doubtful fasciculus, and its fibers are supposed to end in the thalamus and to conduct certain of the touch impulses. The remaining fibers of the anterior funiculus constitute what is termed the anterior proper fasciculus, fasciculus anterior proprius, anterior basis bundle. It consists of a. Longitudinal intersegmental fibers which arise from cells in the gray substance, more especially from those of the medial group of the anterior column, and, after a longer or shorter course, re-enter the gray substance. B. Fibers which cross in the anterior white commissure from the gray substance of the opposite side. End of section 6. Section 7 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information as a volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Spinal Cord Part 3. Fasciculi in the lateral funiculus. 1. Descending fasciculi. A. The lateral cerebrospinal fasciculus. Fasciculus cerebrospinous lateralis, cross-pyramidal tract. Extends throughout the entire length of medulla spinalis, and, on transverse section, appears as an oval area in front of the posterior column and medial to the cerebellospinal. Its fibers arise from the cells in the motor area of the cerebral hemisphere of the opposite side. They pass downward, in company with that of the anterior cerebrospinal fasciculus, through the same side of the brain as that from which they originate, but they cross to the opposite side in the medulla oblongata and descend in the lateral funiculus of the medulla spinalis. It is probable that the fibers of the anterior and lateral corticospinal fasciculus are not related in this direct manner with the cells of the anterior column, but terminate by arborizing around the cells at the base of the posterior column and the cells of Clark's column, which in turn link them to the motor cells in the anterior column, usually of several segments of the cord. In consequence of these interposed neurons, the fibers of the cerebrospinal fasciculi correspond not to individual muscles, but to associated groups of muscles. The anterior and lateral cerebrospinal fasciculi constitute the motor fasciculi of the medulla spinalis and have their origins in the motor cells of the cerebral cortex. They descend through the internal capsule of the cerebrum, traverse the cerebral peduncles and pons, and enter the pyramid of the medulla oblongata. In the lower part of the latter, about 2/3 of them cross in the middle line and run downward in the lateral funiculus as the lateral cerebrospinal fasciculus, while the remaining fibers do not cross in the middle line, but are continued into the same side in the medulla spinalis, where they form the anterior cerebrospinal fasciculus. The fibers of the latter, however, cross the midline in the anterior white commissure, and thus all the motor fibers from one side of the brain ultimately reach the opposite side of the medulla spinalis. The proportion of fibers which cross in the medulla oblongata is not a constant one, and thus the anterior and lateral cerebrospinal fasciculi vary inversely in size. Sometimes the former is absent, and in such cases, it may be presumed that the decussation in the motor fibers in the medulla oblongata has been complete. The fibers in these two fasciculi do not acquire their medullary sheaths until after birth. In some animals, the motor fibers are situated in the posterior funiculus. B. The rubrospinal fasciculus, Monaco, prepyramidal tract, lies on the ventral aspect of the lateral cerebrospinal fasciculus and, on transverse section, appears at a somewhat triangular area. Its fibers descend from the midbrain, where they have their origin in the red nucleus of the tegmentum of the opposite side. Its terminals and collaterals end either directly or indirectly in relationship with the motor cells of the anterior column. The rubrospinal fasciculus is supposed to be concerned with cerebellar reflexes, since its afferent fibers, which pass through the superior cerebellar peduncle, send many collaterals and terminals to the red nucleus. C. The olivospinal fasciculus, Hellwig, arises in the vicinity of the inferior olivary nucleus in the medulla oblongata and is seen only in the cervical region of the medulla spinalis, where it forms a small triangular area at the periphery, close to the most lateral of the anterior nerve roots. Its exact origin and its mode of ending have not yet been definitely made out. 2. Ascending fasciculi. A. The dorsospinocerebellar fasciculus, Fasciculus cerebellospinalis, direct cerebellar tract of Fleischig, is situated at the periphery of the posterior part of the lateral funiculus, and on transverse section appears as a flattened band reaching as far forward as a line drawn transversely through the central canal. Medially, it is in contact with the lateral cerebrospinal fasciculus, behind with the fasciculus of Lissauer. It begins about the level of the second or third lumbar nerve, and increasing in size as it ascends, passes to the vermis of the cerebellum through the inferior peduncle. Its fibers are generally regarded as being formed by the axons of the cells of the dorsal nucleus, Clark's column. They receive their medullary sheaths about the 6th or 7th month of fetal life. Its fibers are supposed to conduct impulses of unconscious muscle sense. The superficial anterior lateral fasciculus, tract of Gowers, consists of 4 fasciculi: the ventral spinocerebellar, the lateral spinothalamic, the spinotectal, and the ventral spinothalamic. B. The ventral spinocerebellar fasciculus, Gowers, skirts the periphery of the lateral funiculus in front of the dorsal spinocerebellar fasciculus. In transverse section, it is shaped somewhat like a comma, the expanded end of which lies in front of the dorsal spinocerebellar fasciculus, while the tail reaches forward into the anterior funiculus. Its fibers come from the same but mostly from the opposite side of the medulla spinalis and cross both in the anterior white commissure and in the gray commissure. They are probably derived from the cells of the dorsal nucleus and from other cells in the posterior column and the intermediate portion of the gray matter. The ventral spinocerebellar fasciculus begins about the level of the third pair of lumbar nerves and can be followed into the medulla oblongata and pons almost to the level of the inferior colliculus, where it crosses over the superior peduncle and then passes backward along its medial border to reach the vermis of the cerebellum. In the pons, it lies along the lateral edge of the lateral lemniscus. Some of its fibers join the dorsospinocerebellar fasciculus at the level of the inferior peduncle and pass with them into the cerebellum. Other fibers are said to continue upward in the dorsolateral part of the tegmentum of the midbrain, probably as far as the thalamus. C. The lateral spinothalamic fasciculus is supposed to come from cells in the dorsal column and the intermediate gray matter whose axons cross in the anterior commissure to the opposite lateral funiculus, where they pass upward on the medial side of the ventral spinocerebellar fasciculus. On reaching the medulla oblongata, They continue in the formatio reticularis near the median fillet and probably terminate in the ventrolateral region of the thalamus. It is supposed to conduct impulses of pain and temperature. The lateral and ventral spinal thalamic fasciculi are sometimes termed the secondary sensory fasciculus or spinal lemniscus. D. The spinotectal fasciculus is supposed to arise in the dorsal column and terminate in the possibly inferior and superior colliculi. It is situated ventral to the lateral spinothalamic fasciculus, but its fibers are more or less intermingled with it. It is also known as the spino-quadrigeminal system of Mott. In the brainstem, the fibers run lateral from the inferior olive, ventrolateral from the superior olives, then ventromedial from the spinal tract of the trigeminal. The fibers come to lie in the medial portion of the lateral lemniscus. E. The fasciculus of Lissauer is a small strand situated in relation to the tip of the posterior column, close to the entrance of the posterior nerve roots. It consists of fine fibers which do not receive their medullary sheaths until towards the close of fetal life. It is usually regarded as being formed by some of the fibers of the posterior nerve roots, which ascend for a short distance in the tract and then enter the posterior column. But since its fibers are myelinated later than those of the posterior nerve roots and do not undergo degeneration in locomotor ataxia, they are probably intersegmental in character. In addition, the fasciculus or tract of Lissauer contains great numbers of fine, non-medullated fibers derived mostly from the dorsal roots, but partly endogenous in origin. These fibers are intimately related to the substantia gelatinosa, which is probably the terminal nucleus. The non-medullated fibers ascend or descend for short distances not exceeding 1 or 2 segments, but most of them enter the substantia gelatinosa at or near the level of their origin. Ransom suggests that these non-medullated fibers and the substantia gelatinosa are concerned with the reflexes associated with pain impulses. F. The lateral proper fasciculus, fasciculus lateralis proprius, lateral basis bundle, constitutes the remainder of the lateral column, and is continuous in front with the anterior proper fasciculus. It consists chiefly of intersegmental fibers which arise from cells in the gray substance, and, after a longer or shorter course, re-enter the gray substance and ramify in it. Some of its fibers are, however, continued upward into the brain under the name of the medial longitudinal fasciculus. Fasciculi in the posterior funiculus. This funiculus comprises 2 main fasciculi, that is, the fasciculus gracilis and the fasciculus cuneatus. These are separated from each other in the cervical and upper thoracic regions by the posterior intermediate septum and consist mostly of ascending fibers derived from posterior nerve roots. The fasciculus gracilis, tract of Gall, is wedge-shaped on transverse section and lies next to the posterior median septum, its base being at the surface of the medulla spinalis and its apex directed towards the posterior gray commissure. It increases in size from below upward and consists of long, thin fibers which are derived from the posterior nerve roots and ascend as far as the medulla oblongata, where they end in the nucleus gracilis. The fasciculus cuneatus, tract of Burdach, is a triangular on transverse section and lies between the fasciculus gracilis and the posterior column, its base corresponding with the surface of the medulla spinalis. Its fibers, larger than those of the fasciculus gracilis, are mostly derived from the same source, that is, the posterior nerve roots. Some ascend only for a short distance in the tract and, entering the gray matter, come in close relationship with the cells of the dorsal nucleus, while others can be traced as far as the medulla oblongata, where they end in the gracil and cuneate nuclei. The fasciculus gracilis and fasciculus cuneatus conduct 1. Impulses of conscious muscle sense. Neurons of the second order, from the nucleus gracilis and nucleus cuneatus, pass in the median lemniscus to the thalamus, and neurons of the third order, from the thalamus to the cerebral cortex. 2. Impulses of unconscious muscle sense, via neurons of the second order from the nucleus gracilis and nucleus cuneatus, pass in the internal and external arcuate fibers of the medulla oblongata to the inferior peduncle and through it to the cerebellum. 3. Impulses of tactile discrimination, via neurons of the second order from the nucleus cuneatus and nucleus gracilis, pass in the median lemniscus to the thalamus. Neurons of the third order pass from the thalamus to the cortex. The posterior proper fasciculus, posterior grand bundle, posterior basis bundle, arises from cells in the posterior column. Their axons bifurcate into ascending and descending branches, which occupy the ventral part of the funiculus close to the gray column. They are intersegmental and run for varying distances, sending off collaterals and terminals to the gray matter. Some descending fibers occupy different parts at different levels. In the cervical and upper thoracic regions, they appear as a comma-shaped fasciculus in the lateral part of the fasciculus cuneatus, the blunt end of the comma being directed towards the posterior gray commissure. In the lower thoracic region, they form a dorsal peripheral band on the posterior surface of the funiculus. In the lumbar region, they are situated by the side of the posterior median septum and appear on section as a semi-elliptical bundle, which, together with the corresponding bundle of the opposite side, forms the oval area of Fleischig. While in the conus medullaris, they assume the form of a triangular strand in the posterior medial part of the fasciculus gracilis. These descending fibers are mainly intersegmental in character and derived from the cells in the posterior columns, but some consist of the descending branches of posterior nerve roots. The comma-shaped fasciculus was supposed to belong to the second category, but against this view is the fact that it does not undergo descending degeneration when the posterior nerve roots are destroyed. As already stated, each spinal nerve possesses two roots, an anterior and a posterior, which are attached to the surface of the medulla spinalis opposite the corresponding column of gray substance. Their fibers become medullated about the 5th month of fetal life. The anterior nerve root, radix anterior, consists of efferent fibers, which are the axons of the nerve cells in the ventral portion of the anterior and lateral columns. A short distance from their origins, these axons are invested by medullary sheaths and, passing forward, emerge in 2 or 3 irregular rows over an area which measures about 3 millimeters in width. The posterior root, radix posterior, comprises some 6 or 8 fasciculi, attached in linear series along the posterior lateral sulcus. It consists of afferent fibers which arise from the nerve cells in the spinal ganglion. Each ganglion cell gives off a single fiber which divides in a T-shaped manner into 2 processes, medial and lateral. The lateral processes extend to the sensory end organs of the skin, muscle, tendons, joints, etc., somatic receptors, and to the sensory end organs of the viscera, visceral receptors. The medial processes of the ganglion cells grow into the medulla spinalis as the posterior roots of the spinal nerves. The posterior nerve root enters the medulla spinalis in 3 chief bundles: medial, intermediate, and lateral. The medial strand passes directly into fasciculus cuneatus. It consists of coarse fibers, which acquire their medullary sheaths about the 5th month of intrauterine life. The intermediate strand consists of coarse fibers which enter the gelatinous substance of Rolando. The lateral is composed of fine fibers, which assume a longitudinal direction in the tract of Lissauer and do not acquire their medullary sheaths until after birth. In addition to these medullated fibers, there are great numbers of non-medullated fibers which enter with the lateral bundle. They are more numerous than the myelinated fibers. They arise from the small cells of the spinal ganglia by T-shaped axons similar to the myelinated. They are distributed with the peripheral nerves chiefly to the skin, and only a few are found in the nerves to the muscles. Having entered the medulla spinalis, all the fibers of the posterior nerve roots divide to ascending and descending branches, and these in their turn give off collaterals which enter the gray substance. The descending fibers are short and soon enter the gray substance. The ascending fibers are grouped into long, short, and intermediate. The long fibers ascend in the fasciculus cuneatus and fasciculus gracilis as far as the medulla oblongata, where they end by arborizing around cells of the cuneate and gracile nuclei. The short fibers run upward for a distance of only 5 or 6 millimeters and enter the gray substance, while the intermediate fibers, after a somewhat longer course, have a similar destination. All fibers entering the gray substance end by arborizing around its nerve cells or the dendrites of cells, those of intermediate length being especially associated with the cells of the dorsal nucleus. The long fibers of the posterior nerve roots pursue an oblique course upward, being situated at first in the lateral part of the fasciculus cuneatus. Higher up, they occupy the middle of this fasciculus, having been displaced by the accession of other entering fibers. While still higher, they ascend in the fasciculus gracilis. The upper cervical fibers do not reach this fasciculus, but are entirely confined to the fasciculus cuneatus. The localization of these fibers is very precise. The sacral nerves lie in the medial part of the fasciculus gracilis and near its periphery, the lumbar nerves lateral to them, the thoracic nerves still more laterally, while the cervical nerves are confined to the fasciculus cuneatus. End of section 7.